Hi, this is the feedback video for the AS mock. So I'm going to go through some questions more than others. Some I'll just read off the mark scheme. So to start off with this first question, it's important you identify what types of molecules each are. So starch, glycogen, deoxyribose are carbohydrates. Deoxyribose because it's a sugar. It's the sugar found in pentose sugar found in DNA. And DNA helicase is an enzyme, which is a protein. So the only things that contain CH and O are the carbs. Protein also contains nitrogen, so that's not right. Protein's the only thing that's made from amino acids. And the only two that are found in both are deoxyribose and DNA helicase, because the DNA found in both and DNA replication happens in both. Really important that you circle the two adjacent hydroxyl groups to remove your water and condensation. Some students missed off this question. I've got no idea why. So just make sure you're not using one of the hydrogens. You have to have your oxygen link for your glycosidic bond. Hydrogen bonds are important in making a cellulose molecule. So what do? Well, they hold the cellulose molecules together, forming microfibrils. That's a mark. And the second mark, the lots of hydrogen bonds, so that makes it strong. Starch is spiral, so it makes it compact for this last mark there. Lots of students going wrong with this question. The idea that a lot of students just wrote down the process of DNA replication without relating each point to how the structure of DNA enables accurate replication. So you can't just say semi-conservative replication. What allows that is the fact that the structure of DNA has two strands. Just two strands of allows semi-conservative replication. The hydrogen bonds which hold the chains together are weak, allowing the cells to separate. That leaves the bases exposed, which can act as templates. It's the bases that act as templates, not just the strands. What happens then? Three DNA nucleotides come along in and you have complementary base pairing. A with T, C with G. And then you've got a new DNA molecule formed with one original strand and one new strand. The next question as well was poorly answered. How the behaviour of the chromosomes produces two genetically identical cells. So first of all, how the cells are genetically identical at the end. Well, first of all, your chromosome, which is two chromatids joined by a centromere, each chromatid is genetically identical. So that's the first thing of how it can lead to genetically identical cells. Also, at the end of in telophase, each pole of the cell will have identical copies of chromatids, identical copies of each chromosome. So that's how it's genetically identical. So then just go through the points of what happens to the chromosomes. So you're not interested in what happens to anything else, like when is the spindle formed and that kind of thing, just what happens to the chromosomes. So prophase, the chromosomes coil up, condense, shorten. And then the chromatids move to the equator, attached to the spindle. Then the spindle fibres contract, so the chromatids move towards opposite poles. And then finally, a nuclear envelope forms around identical sets of chromosomes at each pole. The key thing to answering this question is realising that this will only kill the cancer cells. It will not kill healthy cells. It only kills the cancer cells because they're the ones that have the low oxygen environment. This question was terribly answered. Please go to the mark scheme to have a look at that. The reason for it being terrible is because students were not using the ester bond to join onto your glycerol. So the glycerol will be joined here, and the R group will be here. It is not the R group which joins onto the carbons here. So three fatty acids will be joined, ester bond, here's your glycerol, so that's the carbon for the glycerol, and then you've got the R group here, and that'll happen 3 times. Quite simply for this one, triglycerides are not made from monomers. They're actually made from different units. I would highlight the key things that will give you clues in the question. So the clues are quite simple. This has got a really big triglyceride. So white fat cells are used for storing fat, for storing triglycerides. Brown fat cells have got many mitochondria. What could the advantage of that be? So the mitochondria will release heat energy, warming the animal up. So many mitochondria, so release heat energy to warm the animal up. Students are clearly not revised the structure of a chloroplast. You need to know both the structure of a chloroplast and of a mitochondrion. So this being a chloroplast, each of the individual discs is a thylakoid. The big stacks are grana, so X could be either thylakoid or grana. Y, the thing that looks like the cytoplasm that makes up chloroplast, is not the cytoplasm, it's the stroma. Function of a chloroplast, very basic. I cannot believe students who got this one wrong. So the idea it contains chlorophyll, that wasn't marked, but it's worth putting down anyway, which absorbs light during photosynthesis so that the plant can make glucose, sugars, or starch. Maximum length of the chloroplast. So your calculation, so the actual length is the size of the image divided by the magnification. You have been given the magnification in the question, so you simply need to measure the maximum length, which was 80 millimeters, convert that into micrometers, because in the question your final answer was asked to be in micrometers. So that is 80,000 divided by the magnification of 30,000. That gives you an answer of 2.67 micrometers. Big errors made here with rounding. I had a lot of students having it 2.66 or 2.6. So you would not get both marks. You get the mark for dividing by 30,000, dividing by the magnification, but not the final mark. You must start practicing annotating your graphs and using things like highest, similar, same, lowest. Clearly these three bars here are higher. Every single category is higher than the non-smokers. So the conclusions that you could be making about this, so both, you can see all three bars are going up each time, and between each category it's going up. So both smoking and drinking increases the risk. You could say 20 to 40 cigarettes or over 40 cigarettes increases the risk each time compared to non-smokers. You could say that 40 cigarettes per day and 4 alcoholic drinks are the highest risk of developing mouth cancer. And the other thing as well, if you spotted the anomaly, so on this one here, increasing the number of cigarette smokes for 1 to 3 alcoholic drinks has not increased the risk. And finally, for this one here, you could have inherited a mutation, you could have an environmental factor, for example, passive smoking, diet, age, that kind of thing. Hopefully you can see that the chromatids are moving towards opposite poles, so so it must be anaphase. But how does it make them genetically identical? Each of these chromatids, so this pair, these two are genetically identical. In this pair, these two chromatids are genetically identical. These two chromatids are genetically identical. These two chromatids are genetically identical. So each pole will receive genetically identical copies of chromatids. There were big mistakes made in this question here. Students not saying some cells were this, some cells were that, without clearly stating. So the ones that have got 8.4 units must have gone through DNA replication, or they could be in anaphase, metaphase, prophase, one of those stages in mitosis where the DNA has replicated beforehand and it's not yet divided. The ones that have got 4.2 must either be in G1 of interphase, so before DNA replication, or the cells must have just divided at the end of mitosis to get at 4.2. In meiosis, it halves the amount of DNA, so this is the amount of DNA in a normal cell, so half of that will be 2.1. You must practice pointing out the points for this. It will really help you with questions later on. I'm going to go through question 7 C with you now. It's on the next page, but you need this information to be able to answer it, which is why you shouldn't necessarily accept the conclusion that it treats cancer, bromelain. So for that one, it's only been done on cancer cells. It's not been done on whole living organisms. It's been done on mice. It's not been done on humans. It's only been done on one type of cancer, so you can't make the conclusion that it will treat all types of cancer. And because you've only done it on cells and you've only done it on mice, you cannot predict the effect on humans. We've got a control variable here having the same number of skin tumour cells. So it's usually a 2-mark question for a control variable, the first mark being allows comparison. So please most people put that in anyway. But the second mark is always the most significant. Why is it kept the same? Why is it like comparison? So the intensity of purple could be the same at the start. With this one here, again, not using the graph to answer the question, lost a lot of marks. So why does it support the conclusion that it did not kill cancer and stop them dividing? Well, it stopped them dividing because you can see it's pretty much constant. It's levelled off, so they're not still dividing. And you can say it hasn't killed them because you'd expect the line to decrease if the cancer cells were being killed. So just been through this question on the previous page. How is the rate of the rate of cell division is important in investigations into cancer. Explain why. So it allows you to see whether your treatment's having an effect or not, because the faster the rate of division, the faster the rate the cancer would grow. So it allows you to see how effective your treatment is. Common how science works question here. So with something that's life-threatening, you can't remove a treatment that you know works anyway. Otherwise, it's unethical because the patient could still die, their life would be put at risk. So you have to give both. This question here, there are 2 parts to this question. It's a 2-mark question, and a lot of students just wrote why you push down. So you push down to squash the cells to make them thin enough for light to pass through. You don't move the cover sideways because otherwise you could break the chromosomes, or you could roll the tissue up so you get several layers on top of each other, so that would stop it being thin. With this question here, it's difficult to tell if it's in anaphase or telophase. Actually, it is in telophase, and you can tell because some of these chromatids, like that one there and this one here, they're not all pointing like they're moving away from each other. So the mark points for this. The chromatids are at 2 poles at either end of the cell. So they have reached the poles at either end of the cell. And you know that the second mark, the chromatids have been pulled apart, so it can't be metaphase, because they've got this classic V shape. For this question here, you need to work out the proportion of cells that are in anaphase. So 6 divided by 200 is equal to 0.03. The number of cells is also proportional to the amount of time that it spends in that stage. So you need to times that by, which will be 960, which is the number of minutes in 16 hours. So you got that from doing 16 times 60. So 0.03 times 960, that will give you an answer of 28.8, or simply 29 minutes. I'd like you to look at the mark scheme for this question. It's quite a straightforward one to go through. Just want to learn a sequence of events. There's things where people fell down was not explaining. So why do you use isotonic solution? So to stop osmotic damage to the organelles. Why do you use a buffer? To stop the enzymes denaturing in the organelles. And the final step as well, how you finally get the mitochondria. You've already moved the nuclei to lower speed. You have to say you use a higher speed to centrifuge, and where you'd find the mitochondria, you'd find it in the pellet at the bottom. Okay, so the couple of things there, not explaining the answers fully. Principles and limitations. A lot of students weren't sure what the word principles meant, but it's kind of like how it works. So the idea that a beam of electrons passes through the sample. The darker the colour is because they've absorbed more electrons. And why electron microscopes are really good is they've got really high resolution because they've got a very shorter wavelength than light. Electrons have got a shorter wavelength than light. for the limitations, it's just a list you need to learn. Can you please look at the mark scheme for that? So this is the last question. I'm going to do bits for the very last question as we go through, 11C, where you evaluate whether you should give both drugs at the same time. So as we pick out the information to answer that question, I'll highlight what you should be annotating on your work. So things for the very last question, the evaluation, what can we get from this? So first of all, it's done on mice, so they are not humans. So you cannot predict the effects for that. It's only done on 10 mice, so we can say that's not representative. Sorry, my writing's terrible. So only done on 10 mice, so it's not representative. Right, we're going to carry on with the normal questions, and we get more information for the last question. We'll do it as we go through. This question here, so why did they remove the ovaries? Well, that was the main source of estrogen. So that means that they allow, by adding the substrate allows you to control how much estrogen is produced. So the only estrogen that is being produced is due to the amount of aromatase that you add. For this last question here, a lot of students were just putting something like, and when I move up, you'll think this answer sounds great, and then I'll move the answer up. So the enzyme is to make estrogen is inhibited, and fulvestrant binds to the receptor. That sounds really good, but then you scroll up. Right, in the question, it has told you that this inhibits the enzyme and this binds to the estrogen receptor. You've not told me what the significance of those points are. So for question B, it inhibits the enzyme aromatase, so less estrogen is produced. It binds to the estrogen receptor so that estrogen cannot bind to the receptor. So this is kind of a diagram that's going on here. You've got the substrate which you add, catalyzed by aromatase, produces estrogen. That binds to a receptor and causes cancer in the cells. So if you reduce the amount of estrogen that's being made by inhibiting aromatase, or if you block the receptor using fulvestrant, then it's going to reduce the amount of cancer that you get. Things that we should have been annotating on here. So again, this is to help the very last, the evaluation question. That's the only question left to go now. So only done for 15 weeks, the final results. So you could not necessarily see the maximum effect of the drug. We can see that there are the least number of tumours with both groups, much fewer than all the other groups. We can also see that it has the lowest mean compared to these three, so the lowest mean area of tumour, so the tumours are smaller. We've also got the lowest top range. So that's a good point, so 2 good points there, lowest mean, lowest top range. Why you should, that's the reasons for giving both. But the mean is similar. It is similar to the other ones, so that's a reason against. Also, the ranges overlap. So that means in some situations, actually quite a few situations, because there's a lot of overlap between that bar and these bars, quite often the effect of this drug was the same as the effect of these drugs. But you can't prove any significance with this because we've used a range rather than standard deviation. Standard deviation would have been better. So one other point, the only other point, so we had some information from the first where we highlighted the text, we went through it, we've got the graph here, and the only other point you could have is there might be side effects which you've not studied. We've been through the points for that. If you want a recap, please look at the mark scheme. I'm going to go through the mark scheme now, just show you them. So this, I don't know why, but in the printout we got it was wrong. Just remove that row. So you've got the top row 3 ticks, second row only 1 tick, bottom row 2 ticks for deoxyribose and DNA helicase. There we go. Any questions, please ask.